Thank you for coming. Thank you for braving the cold weather, avoiding, watching the football till later, even though England are out, so that's fine. And train strikes, etc. I had to come on Monday, and I went on the train so I can go home tomorrow, and I cancelled 3 trains. I went to the train station 4 times, and then the 4th time I was on a train, thank goodness. So firstly, I want to say thank you to all the people that have taken part in this fertility series, Fertility in the 21st Century. We've had quite a different range of disciplines and discussions around fertility. And if some of you have been at all the lectures, you will recognize some of the names. So we started off with the biology and then the biochemistry. And then there was Professor Claire Lynch, who's been through IVF with her partner. And she was, she's a creative writer, so she was talking about that. And then we had sperm health quite recently on the 6th with the 2 leading experts, one here from Brunel, Andreas Kortenkamp, and then we had Sarah Martins de Silva that was live from Dundee talking about sperm health and the effect of the environment on that. And then the discussion that we had last night with our psychologists, the sociologists, and the law professors around having it all, choosing career and a family. And tonight, I will just give an overview of some of the things that are happening for fertility just now that are sort of breakthroughs, but also what might be coming. A lot of these issues will obviously have to have some robust ethics and discussions with lawyers around them, but are still interesting and possible from a biological point of view. So we'll have a look at those. And then we're followed by 2 very interesting programs that are run in the center that I direct, and that's the Center for Genome Engineering and Maintenance. We call it CenGEM because we really like that snappy name. That's in biosciences, in the College of Health, Medicine, and Life Sciences. And these talks are coming from 2 very exciting scientists to understand how abnormalities arise in specific cell types in earlier embryogenesis using an in vitro stem cell model. And that's Dr. Christina Pina here. And she's also a hero of coming here tonight because she flew in from a big meeting in America this morning. She landed at Heathrow this morning, came to work, then went to graduation up in central London, and now has come back on the tube to be here. And we are sending her home in a taxi because she's done enough. So thank you, Christina. And then to develop the safe gene therapies to treat specific diseases in the fetus in the uterus. And that's, we have Professor Mike Themis here who is working on that. So Christina is a senior lecturer and a qualified medic. She trained in Portugal for her medical degree. She is the head of the Cancer Mechanisms Cluster and works on early childhood leukemias. Mike is a professor of gene therapy and toxicology, and he is the founding director of Testavet Limited that's established here through his work at Brunel. But first of all, I will just give a quick overview of some of the things that I found while going through looking at the future of fertility. So one of the things I was going to talk about is that, and it came up yesterday evening, didn't it, was this elective egg freezing. So it's very possible now to freeze one's eggs, and you can pay to have it done. And there are even companies like Google and Facebook that offer it. So it's part of the perks when you start working with them. And there was then this discussion about, okay, it's a good offer, but it shouldn't be taken as it's an absolute. It's not 100% guarantee that you will then be able to use those eggs to create live births. So I think the rates are quite good. And I know from 16 years old, they talk about, the girls at this age talk about maybe they'll freeze their eggs so that they can go on and have their career and maybe not suffer some of the issues that their sort of older mothers or older generations have gone through getting older and missing that biological window. So, so that was a discussion that came up yesterday. So there's ways of doing it, and what you need to do is prevent ice crystals forming. And so you don't want those ice crystals to damage the egg coat or even inside the egg. So there are very careful ways of doing that. And for a long time, it's been a slow freeze, very slow, take it very down. And this is what we do for our cells when we're working in the laboratories, freeze them very slowly, and we thaw them rapidly. But there has been some recent developments where we have rapid freezing, where even the freezing medium, which is liquid nitrogen, is injected into the egg to make it freeze very rapidly. So the success rates for live births from frozen eggs are between 50 and 75%. But there's a caveat with that. It depends then on the age of the female when she donated the eggs to be frozen, okay, because there's still that aging aspect to within the ovary, okay, and the same issue with getting older eggs, same when you freeze them down, okay, and also then the freezing procedure, so that needs to be followed very well, precisely, but also sometimes then the clinic or the people that are dealing with the frozen eggs, different individuals can have different success rates. There's no reported increase in genetic abnormalities from the frozen eggs above non-frozen eggs, so that's good. And quite often the eggs are tested prior to being used for IVF for their viability, which is not always the case when they're harvested for IVF. So they are studied beforehand. But also ovary tissue can be frozen and grafted into individuals, and it becomes functional. So the ovary tissue can go into transplanted into somebody else, and then within a few months there's menstruation, so eggs are developing and passing out of the ovaries. And I think we've known for a long time that you can freeze sperm quite easily, but then also cells, the cells that precede sperm. So we looked at spermatogenesis in the very first lecture, the different cells, and so they can be frozen and then developed in vitro afterwards, and even sperm stem cells can be frozen and then differentiated in culture. It's also possible to do in vitro egg maturation, so taking sort of earlier oocytes, so not oocytes that have matured, so eggs that are immature, and collect them and then mature them in culture. So they get put in a dish without serum, but they get the hormones in the dish, which means that individuals that are not going to do very well with these very high levels of hormones that are given for ovarian stimulation, and that might be because they have PCOS or they have some cancer that will not respond very well to having a lot of hormone pumped into the body. So these immature oocytes can be collected and then matured over 24 to 48 hours and then used in IVF. And so there's no evidence of increased levels of congenital birth defects when using these matured oocytes that have been done that in culture, and the miscarriage rate is no greater than it would be normally. And so using a certain stage of maturity of the oocytes, or immaturity, should I say, the success rates are quite high. But now even some very, very immature oocytes can be collected, and there have been some live births from them, but the rates are not quite the same as they are for collecting the later, but still immature oocytes. Third parent, IVF, which works with nuclear transfer. So this does come from this methodology that was developed. And this gives me an excuse to show my young self with the real Dolly the sheep. So Dolly the sheep was at Roslin and was made by a dear friend of mine who's now passed on, but Keith Campbell. So I show this picture whenever I can, don't I, Christina? Because it's one of my favorites. So, but This Dolly the sheep was made possible by taking an adult cell nucleus and putting that into an enucleated egg. So the enucleated egg is where a nucleus has been taken out, and then the adult nucleus was put in. So that's what was happening there. But this is not cloning, okay? So what happens here is that maybe the mother who wants to become pregnant has a mitochondrial disease, and so that's preventing conception, etc. But what can happen is then there's a donor egg where the nucleus, so the main part of the DNA, the genome, is taken away. Okay, so it's enucleated, and then the fertilized, the nucleus from the fertilized egg, so that it's got the male and the female genomes together, is then put in that egg. And why it's called a third parent is because the mitochondrial DNA comes from the other female. So that stays there. And there's 37 genes in the mitochondria that are required for us to develop. And so that's why it's called the third parent IVF. It's possible to have womb transplants. So individuals have been having, not very many across the world, but there have been whole womb transplants. And it's possible to be born without a womb, or you lose it through disease. It has to be removed, sort of early cancers and that. So the first baby that was delivered from a womb transplant was in Sweden in 2014, and then there have been others around the world. But these data here talk about, out of 33 women that received transplanted wombs, 27 became pregnant and bore children. So that's quite a nice high percentage there. And also, one of these mothers is now going for her second child, and then it was in a newspaper, a Newsweek or something, will have that womb removed afterwards. There is research ongoing to do womb transplants into transgender women. It does happen, but they're not functional. And doing the transplantation of the womb is fine, and the ligaments can grow, but it seems to be it's the pH and the tissue that's used to build the vagina is an issue for fertility. There are up-and-coming artificial wombs, and people are talking about it for humans, but it's obviously not been used for humans yet. So this is where it's in the future, where couples have the uterus is compromised or not present, and it would allow fetal surgery more easily. And it hasn't, as I say, it hasn't been done for humans yet, because a human embryo cannot be allowed to exist past 14 days in culture. But it has been done for lambs, and it has been done for goats and mice. Okay, so this is a cartoon here of where premature lambs were put in the artificial wombs for 4 weeks and continued developing. But the heart is beating in the lamb, and that's what's creating the circulation. What about making new gametes? So to make new sperm and egg, there are quite a lot of different studies doing this, and I think this is where there needs to be the robust ethical discussions, and the lawyers need to also be involved, because so there was a study that I think you find it quite easily if you go on Google, as I did. Stem cells found in mouse and human ovaries that give rise to ova and eggs. So take these stem cells out, you can make fresh eggs. But that was proven quite recently to not be correct. So there are some Japanese scientists that have taken blood cells, so they're taking adult cells, and you can make these induced pluripotent stem cells. So these cells get sort of de-differentiated, reprogrammed back to a much earlier sort of state, more like an embryonic stem cell. Okay, and then those have been able to be made into immature eggs. Now, this causes some people a concern that you could just maybe get an adult cell off George Clooney or something, and then go and get yourself a George Clooney baby. But I mean, that's what the press talk about online and stuff. But so that hasn't been progressed for humans. there's a lot of discussion about being able to combine two female genomes, and there is some success in this in mice, but it means that you have to alter the imprinting. So one of combining one of the eggs, you need to reorganize its genome and to change the epigenetics in that for it to make it possible. And so there's quite a lot of discussions that that's probably going to be too difficult, both biologically and ethically, to do that for humans. Mouse embryonic stem cells have been induced to become primordial germ cells, PCGs, that we find in early embryos, and then they are the precursor cells to sperm and eggs. And so if you can make them, then you can go along and differentiate in culture and make sperm and eggs. And sperm produced from these PCGs have fertilized mouse eggs. Embryos have been transplanted, pups born that were fertile. So that has happened in mice, but again, that step to humans wouldn't happen. Okay, and then recently rat sperm. So a lot of work has been done on mice. Now rat sperm, and it's an article from this year, has also been induced from stem cells. And so sort of changing cells to make new gametes. AI is also being used. So you can train AI to see where you have a healthy egg, a healthy embryo, and then take lots of time-lapse images that go into the system, but also then what happens afterwards. You know, what happens with that egg, that embryo, what was the outcome? And so that keeps teaching the AI how to predict whether there's going to be a healthy full-term pregnancy or not from a certain egg or a certain sperm or embryos. And presently, there's an increasing accuracy using AI, and this is Violet. Violet is able to do better by 18% than expert embryologists.